Uh, First, I just want to welcome everybody. I see that people are still joining, and we have a wonderful turnout today, which is really great to see that there's so much engagement and interest from the NM community, um, which is such an important part of what we're doing with AFBS. So I just first want to just welcome you all here today. I'm Jen Tozer. I'm the Executive Director of Foundation Building Strength, and I am truly honored to be here today. Um, Let's see. I want to start with an overview of today's session. We'll just do an overview of a foundation building strength because this webinar and the research that we're going to be sharing today is all brought to you by a foundation building strength and the funding that we do and this is what our priority is. So I want to go over our research, a brief overview of our research priorities. And then introduce our researcher presentations or presenters, and then get started with their presentations, followed by Q&A. So first, as I said, this is brought to you by a foundation building strength. AFBS was established in 2018 or 2008, excuse me, to support NM families with the goal of finding treatments for Nemaline myopathy. That is always our primary goal and will continue to be, and that's why we're here today to share about the research. But we also want to support you all in the community now as we work towards treatments. So we have a really great dynamic of partnerships with our researchers and clinicians, and also within the community to support families and affected individuals with Nemaline myopathy now. So just as a reminder of our mission, it's to support Nemaline myopathy treatments, to find Nemaline myopathy treatments. And right now is a really exciting time for AFBS because we have just recently granted new grantees to support their research, and some of them are on here today. So we're so excited to get to that. AFBS has committed to investing $3.6 million over the next 3 years to support our research priorities, which is gene therapy approaches, small molecule drug therapies, and supporting natural history studies to help us get to human clinical trials, an essential step. So this is a big momentous moment or time for AFBS and for the NM community to start accelerating our research progress even further and faster. And I just want to emphasize that 100% of all donations go directly to research. We have all of our overhead costs are covered by our board of directors and our pillars of strength donors. So all of our donor support goes to research. In addition to the research, like I said, we are focused on funding research and finding treatments for NM. But in the meantime, we want to support the communities. We want to support you all. And we want you to be involved in what we're doing, because without you, we won't be able to make progress. So I've listed on this slide a link to our website that outlines ways for you to get involved, whether that's connecting on a support group or getting involved in advancing research through joining our registry platform, or joining the natural history study that Tina will be sharing about soon, getting all of the resources that we have available to support your care now, and then also fundraising, which is an essential part of what AFBS does, because without donations, we wouldn't be able to support this incredible research and make this progress that we're making. So just check out the website, and if you have any questions, please reach out. We're always here and available to help and support you in any questions or anything that you need. But I know why we're all here today. It's to hear from these incredible presenters and researchers and clinicians, to hear about the latest in NM research and progress towards clinical trials. So it is an honor and a privilege to have each of these individuals joining us today. Thank you so much for taking your time to be with all of us. We have Tina Duong from Stanford, who will be talking about the upcoming NM Natural History Study. Dr. David Mack from Washington University, speaking about 3D engineered muscle tissue. And then Dr. Afrooz Rashnonejad, who is focused on gene therapy for ACTA-1 patients. The way that this will be set up is we will have our first presenter, Tina, who will share about the natural history study, and then that will be followed up with a Q&A. So we want to encourage you to put your questions in the chat. I'll be monitoring the chat, and then we'll be helping to deliver those questions to Tina at the end, and then we'll move on to Dr. Mack following. So if possible, just start with questions about the natural history study, but of course, enter any questions that you have. We want to be able to get to as many or as all if possible. And just use that chat feature, and everybody is already on mute. So I appreciate that, because that's an important part of being able to get through this time. And again, this is a 90-minute session. So we want to be able to use all of this time to support your questions and to hear from our wonderful presenters. So that being said, I would love to pass it over to Dr. Tina Duong to start her presentation. Oh, and I'm just seeing in the chat, this is being recorded, and this will be shared after the session. So this will be available for anybody who's unable to join. If you want to share it with anybody else, this will be available soon.
Great. Thanks, Jen. Are you able to hear me okay?
Yeah.
Okay, great. Hi, everyone. I'm Tina Duong, and I'm working on the natural history study alongside Dr. 𝗕𝗄𝗄, and we're at Stanford University. First, I kind of want to talk about natural histories in general and why we need it in the landscape of drug development or looking for treatment. Then I'll introduce a quick introduction about the study and the study protocol, so you know what to expect, as well as timelines, and if you're interested, some of the study contacts that you can reach out to. So first, we'll start off with clinical trial readiness. So why do we do it? It's really our roadmap that allows us to assess the characteristics of Nemaline myopathy when there is no treatment. And we know that these characteristics change over time, so different clinical presentations. And to really truly understand if a treatment is working, we really have to know where we're starting from. And this is what natural history studies do. They not only study the characteristics of a clinical of presentation, but also they build the bridge. So when there's an idea in the lab and it needs to be tested in a clinical trial, everything in between really matters for a successful trial. And I'll tell you a little bit about some of the components that are essential that we've learned from other neuromuscular diseases that have had successful treatments on how to do this better and more efficiently. So natural history studies really are the foundation of future research. It's usually the point of comparison. A lot of times you'll hear in research studies where they reference placebo or a control cohort. These are the things that allow people to, researchers and clinicians, to understand what the difference is from no treatment and treatment. And there's multiple ways that we can do that. And it also allows a different way to even design trials that may be more efficient, that makes more sense for individuals with a rare disease. It also helps us find key endpoints, because if you can't measure it, how do you know it changed, right? And so depending on the treatment, whether it's a phase 1, 2, or 3, we may want something like a biomarker that is sensitive to change in the early trial, so we know that it's working. And then while we're doing that, we have to know when we get to a larger phase trial, which is a phase 3, that what are we going to measure that's going to be clinically important? And so we learn all of this along the way. And so by the time there is treatment availability, we know how to design a trial that will give us the best approach to show its effectiveness. So clinical trial readiness is definitely a team. I think the most important thing here is to make sure that the patient voice is heard, because you're the one that's going to tell us what is important to measure. And for the regulatory authorities, clinical meaningfulness comes from patients, right? And so we want to have a dynamic, ongoing dialogue with patients while we're studying this to ensure that we are looking at the correct things. And of course, it's some of the key players that we have to keep in mind in the trajectory or the timeline of clinical trials are, from discovery all the way to access, right? And so we want to make sure that we design trials that allows us to take down the barriers, whether it's at the time of the regulatory approval or it's afterwards to get the treatment through payers. And so with this, we want to make sure that as these therapies are being discovered, we want to know what to measure, how to measure it, and how it changes in the natural history. Because what we want is to have a really promising drug that that is not able to show the impact because we measured the wrong thing. Okay. And the other benefits of a natural history is that it allows us to have more community engagement and awareness. So as we're learning new information, we bring it back to the community. We're able to better understand and really improve the standards of care and guide clinical care a little bit better. And then again, like I said, most importantly, we get the patient perspective on the trial process. But not only that, it's really important for you to give us feedback on the burden of participating in a trial, because that does not go without extra time and effort dedicated by you and your family. There are some differences in research and clinical care. They should go hand in hand, but there's also a piece where you have to be taken care of by your clinicians. The research that we do in a natural history study and for clinical trial readiness is done in a way similar to clinical trials so that we can compare it. So we have to do it in a rigorous, standardized way to reduce that variability. And with this data collection, we have quality control as well as talking to regulatory authorities to understand the guidance there. We need to determine the best tool to measure, as well as look at what kind of genotype or phenotype correlations can we make between some of these patterns that may help with the inclusion criteria for clinical trials. In your clinical care, what it could help is that we could improve the diagnostic process and recognize earlier signs or common signs associated with Nemaline. And then prognostic clarity, helping distinguish from different forms, whether it's the congenital or the later onset. And of course, I think one of the other things is the rate of progression. In rare diseases, what impact is not only a change from baseline to whatever the endpoint is, it may be that we are impacting how fast or slow your disease progresses. And now I'll introduce the natural history study. I think we've all decided that it's going to be called NEMO, and it stands for Nemaline Myopathy Clinical Research Network for Natural History and Outcomes. Our primary objective here is to evaluate the natural history of Nemaline. Our secondary objective is to find measurable outcomes that are reliable, valid, and shows minimal detectable change. These are the basic psychometrics that are required for us to prove that an outcome is effective. And so these endpoints are going to be specific to age because kids are growing. We have to make sure that it also has an explanation from a norm reference on developmental scales. So there are a few of these motor assessments I'll talk to you about in a little bit. But this is one of our team meetings, one of our initial meetings at the MDA, and this is just a few of the members of this team. So to do this, a lot of pre-work occurred. So we had workshops to really look at the objectives. We wanted to harmonize our data in a multi-site way and support regulatory alignment, right? And so to do this, we worked as a group alongside the US as well as the UK group who's also doing this, developed the inclusion criteria that for this study, and then we harmonized on the outcome measures. So what that means is that the US group as well as the UK group and all of us are doing the same thing at the same way so that we can compare this. And then our plan is to share, of course, this information for improved collaboration, de-risk this for drug development companies in order for them to feel invested in Nemaline, and of course, talk to the regulatory agencies and payers. So here's our study design. We're going to enroll 50 children from the ages of 0 to 18 across North America. We're hoping to enroll 5 to 10 per site that we have. And just keep in mind, this is natural history. I think, it's nice as a patient, you may get to learn kind of the clinical trial process as well, participating in these kinds of studies. And then you'll receive your usual standard of care by your treating physician, okay? For how many times you may be seeing us. Your participation is going to be 2 to 4 visits, depending on how old you are or your child is. So visits are every 3 months for individuals who are less than 12. And those who are older than 12, we'll see them every 6 months. Okay. And really, we try to have very few exclusions because we will really want to capture the breadth of everyone who has this disease. So how long are these visits? They could be 1 to 2 days because we're looking at a lot of things. And here are just some examples of what we're assessing. The first one's motor function test, which is, you're walking 10 meters, getting up out of the chair, stairs, and just bed mobility allows us to make a connection to the things that you do at home and your activities of daily living. On top of it, more recently, thanks to AFBS, we're also going to add video analysis of your movement and tracking how much physical activity. Because we know that sometimes the time that it takes for you to walk 10 meters may not change. However, the compensation patterns or the way you use your muscles are different, and we want to be able to detect that because that may be more sensitive to change. Alongside that, we'll also take MRI and ultrasound so we can understand the architecture of your heart and muscle, respiratory assessments to test your breathing, as well as blood collections, questionnaires, and optional skin biopsies for us for further development of clinical trials. So here are the participating sites. Stanford's the leading site for the U.S. network. So depending on where you are and where you're closest to, you can go to any of these sites in California, Dallas, Memphis, Tennessee, Bethesda, Maryland, or D.C. area, Boston, and Toronto, Canada. We also work closely with our colleagues in Brazil, and they also are using the same protocol. This program was developed in combination with the United Kingdom network, and so we share the same database, the same data dictionary, and the same protocol. And so then when we get together, we have a larger collective database than we would alone. Here's our basic timeline. So we're a little bit off of our timeline, so I adjusted it. We're hoping to really at least get someone in before the end of the year. It's a 3-year data collection after we get the first patient in. And so recruitment for this is going to be between December 2025 to September 2029. In between this time, we'll do some interim analysis so that we can give a report to the community as well as AFBS on some of our findings. And then we're hoping by the end of 2029 that we'll be able to share the reports and publish on some of our findings. Here are the study contacts. Of course, and Jen will send this out, so you don't have to take a picture of it, but these are the individuals that you should reach out to if you're interested or you know someone who'd be interested in participating in this study. With that, I want to thank you in advance because if it wasn't for the community, we really would not be able to do these clinical trials and trial readiness. So I appreciate all of your participation and look forward to any questions you have.
Thank you so much, Tina. You really know how to cover a lot of information in a short amount of time. That was fantastic. Thank you. I do want to open it up to questions. I don't see any in the chat. So if anyone has any questions, please just feel free to say them. Lynn, go ahead.
I know we've talked about this before, but we have quite a large population that's in that 17, 18-year-old range. Can you speak about whether an 18-year-old can start with it and how that would work for over the 3-4 years?
Yes, absolutely. So as long as you're 18 or younger, you can enroll in the study. So that means we'll capture you at 18, 19, and 20, but that's absolutely okay.
And Tina, can you speak to, I know there's some that are at early adult ages, like why we've stopped it at 18, just to clarify that for folks?
So we really did that to synchronize with both of the networks, and to there's only so many, to tell you the truth, how many people we could enroll because of a budget, and so we want to make sure that we hone in on the younger population before we went to an older population, and with more funding, we definitely want to make this more inclusive of individuals who are older.
All right, I'm seeing in the chat, our enrolled patients include, are we including all known NM genes, or are we focused on NEB and ACT1?
Right now, we're focusing on NEB and ACT1, and to tell you the truth, originally our protocol was much broader, but like I said, I think we're going to start out with NEB and ACT1 and younger individuals because of funding. Otherwise, we may get a whole bunch of people, and we're not able to connect some of the dots on the clinical.
Makes sense. Thanks.
Yeah.
Okay, and so there's Caroline is sharing that her daughter is part of the UK study, but it's open to all ages.
Yeah, the UK study, they do it, there's components of it. They have some where they're just collecting clinical data, so it's a really small subset. And that's something I think, we could consider as well, a minimal data set for individuals who are older. And so that's why they enroll you can be any age to enroll, but the intense part of the natural history for clinical trial readiness is in the younger cohort.
Thank you, Tina. And it seems there's a study in Belgium. Is this connected? Is this one of our partners?
Belgium isn't one of our partners. I know that our UK partner works closely with Belgium so they may be a similar group.
Yes, actually,
I don't know. Oh,
the PI in the UK also is from Belgium, and he has a hospital, an appointment in Belgium. So I think it's related to that. I cannot confirm, but I think it's related to that.
Yeah, this would be my guess as well, because he actually travels to Poland, Belgium. He's all over the place.
Thanks, Gus. And Gus is our scientific director, so he'll also be on here to be able to answer questions. So grateful to have Gus here. Another question about, so in terms of getting admitted into the study, Karen is asking about if she reaches out to her doctor in charge at the hospital, is it likely that they would be accepted into the study? She has a baby who has TPM2. Would they be a candidate? And is there a stipend to participate?
Really great questions. Depending on where your hospital is, I would actually suggest, I'm sorry, I'm getting over a cold, suggest to reach out to the contacts that I had on the last page for the site that's closest to you, because this is a disease. Not everyone knows that there's a natural history. So please do spread the word, and we're happy to talk to your clinicians if and see how feasible it is. We do have a stipend in the protocol, but for travel for people depending on how far you live, and so we did put into the budget.
Great, thanks, Tina. So there's the question, I think there's questions about the gene. So there's TPM2 and then TNNT1. I think you can speak to that again. I think right now we're starting with NEB and ACT1.
But gosh, Alan up in Boston are still Alan Beggs is still collecting blood from individuals with all subtypes.
Which is a great reminder that even if you can't participate in a study, there are other ways to be involved in research, like tissue donations with, like Tina just said, with Dr. Beggs at Boston Children's Hospital, and other. We have a digital retrospective natural history study that you can also be a part of through Citizen Health. All of that is available on our website. those are nice alternatives if it doesn't fit for the gene or age right now. And so I love to see all the interest, the global interest in these studies. So is there a study in Italy?
Not right now.
Yeah.
But I think once we get momentum, what I've seen is that we start to collaborate, right? And so then we have a protocol, and it makes it easier for other places to start up. So if you have PIs in these countries that you would like us to connect with, we are more than happy to share our protocol and our manuals to ensure that what they're collecting is going to be consistent to how we are collecting it, so it's comparable.
Yeah, and one more thing, to set up all these natural history studies, we had an important meeting in 2019. There was in the Netherlands, was a workshop including many different representatives from different countries, with the idea of making this natural history study as global as possible. And so there are many countries some of them are looking for funding to initiate these clinical trials, this natural history study.
And all that said, I'm a big fan of learn from every patient. And so if you're in clinic and you're seeing your clinicians, I think it also would be helpful if, if there's we could probably propose some kind of minimal data set so that we at least have a measure that everybody's measured consistently, whether you're in a trial, a natural history study or not, so that, it coalesces how we're able to look at care.
Let's see. I'm seeing a question about citizen health from Adrian. So I'll talk just for a minute about citizen health, as we do have it's a retrospective natural history study that we're working on compiling. Citizen health collects patient records, all of your medical records. You just put in where you've been seen, and they get all of the records, upload them, and then ultimately that will be used with your consent to develop a retrospective natural history study based on your medical records. That feature of being able to access medical records through Citizen Health is only available in the United States because of regulatory restrictions. But you can still participate if you're outside of the US. You just need to upload your own medical records, and then they'll be able to be utilized in that way. But because of the just legal restrictions, we can't, they aren't able to access anything outside of the United States right now, hopefully in the future. But right now it's only available in the US.
And that actually triggers me to just mention something as well. The UK and us are working on a data federation as well. And what that allows from a regulatory perspective is for us to share information that it doesn't have your private information in it, and it gives a little bit more breadth. And so hopefully when we, as we're doing this, we can get other people to join where there's not that regulatory risk of private information being shared across country lines.
Okay, so Stacey's asking, my 8-year-old ACTA-1 is currently part of a study at NIH. Is this the same study or a new one?
I mean, I know NIH is part of our study. Gus, do you know? I know that they've done some natural histories, but it may be...
Yeah, they have many different protocols. So yeah, but it's not strictly using this protocol for this natural history study. But I'm sure they will roll the case when they initiate the natural history study with this specific protocol.
Stacy, is that the one with 𝗏𝗏𝗏𝗏?
Originally it was. I'm not sure if Carson. Mom,
yeah, he's got so much going on. He often starts these studies and then gives them off to other individuals. But I mean, he's kind of the main.
He's who we saw first when Wyatt was 1, and then when we went back 2 summers ago. We did not see him. He was mentioned. It was very heavy cardio and pulmonology.
Yeah, I'm as Gus said, I'm
Initially, Dr. Bonneman had come to Philadelphia when Wyatt was a baby, and then we saw him at the NIH as well.
yeah, I'm also confident that that will just be rolled into this.
Okay. Okay.
Now, as Gus said, the readouts might be a little different, so there may be some backtracking or having to figure out how to mesh the previous data with this new protocol, but it'll be part of it. I'm pretty confident.
Okay, thank you.
And that was something we considered in the design of the protocol and the manual and methods as well, so that they in all of these mesh together.
Tina, could you speak to this question from Crystal about will this study help with TNNT1?
Absolutely. I think there's a lot of learnings here. When you're looking at functional measures or motor measures, that goes across the board. That's not, specific to a specific mutation. And so I think that when we learn this, it kind of gives TNT1 a head start in a sense, in regards to understanding how to develop a natural history study and give better direction for wearables, digital health, outcomes, etc.
Great, thank you so much. I think we covered all of the questions. Is there anything else? Drug trials. Okay, well, you know what? Let's get Arash, I would like to address your question, but let's do it after we hear from some of our researchers and their latest work in gene therapy and small molecule developments, and then we can ask I would like to address those broader questions. So I think on that note, I'd like to pass it over to Dr. Mack.
Okay. All right, what are you guys seeing?
Right now, we're seeing your PowerPoint in its original form.
Okay, how about now?
Now that looks great.
All right. Well, great to be with you this morning. I just want to express my gratitude and appreciation for my lab and our collaborators being part of this community. Since teaming up with you guys probably 4 years ago or so, it's been a really fun experience, and I'm just always taken by how dedicated everyone is and how much this team plays really well with each other. So what I want to do today is tell you about our efforts to create 3D engineered muscle tissues from human cells. And here are my disclosures. There's a big effort across the country now to try to improve these organoid or tissue systems. So the NIH started this initiative called the Standardized Organoid Modeling Center, and they put in $87 million. Exactly how those monies will be distributed and who will benefit from them is still a little bit up in the air. But it makes really clear that there's been a widespread acceptance that animal models still have their wonderful utility. They will remain a central part of what we do, but we need to do better. In instances where the animal models don't recapitulate the disease in humans, we need to put major efforts into designing, building, validating, standardizing these engineered muscle tissues or engineered tissues across the body to do drug discovery and validation. It's clear that current models don't have the predictive capacity that we really need them to have. And what I mean by that is, if it works in an organoid, can you predict whether or not it's going to work in the patient? So this is going to be a major effort over the next 5 to 10 years, and we're happy to be a part of it. So how do we make these engineered tissues in my lab? So the idea in general goes something like this. So a patient comes into the clinic, we collect their urine, and in that urine are these sloughed, we think they're kidney nephron cells that we can then put on the dish, expand, and that captures that patient's genotype. Then we use standard, they're called Yamanaka factors, this won the Nobel Prize a few years ago, to turn those urine-derived cells into induced pluripotent stem cells. Those cells have the capacity to differentiate into any tissue in the body. We use them to make cardiomyocytes, skeletal muscle, and we're also getting pretty good at making motor neurons. The real challenge then is, can we take those defective mutant skeletal muscle cells and uncover the underlying molecular drivers of the pathology? Can we trace from mutation to all the downstream effects and how that manifests in a disease phenotype, weakness, muscle weakness, for example, changes in contraction and relaxation kinetics? Can we capture that in a way that we can then, after treatment, we look for the correction of that? So restoring that defective cellular function back toward normal. So that's what we're trying to do for multiple different diseases, and ACTA1 mutant Nemaline myopathy is a big part of the lab right now. So what I want to do today is just show you some of the preliminary data that we've generated over the years. We're not quite to steps 7 and 8, which is taking our best candidates and putting those back through our typical drug discovery validation pipeline, and then with the hopes of giving those back to the patient. We're not there yet. We're kind of just working at step 6. Okay, so I'm going to glaze over this pretty fast. There's a lot of detail here, and I'm glad to talk more in detail about it in the discussion. But we're getting pretty good at making skeletal muscle. The way we do that is to push these pluripotent cells in the upper left, to make them to push them through these choices of lineage bifurcations, to push them toward axial and limb muscles. The way we do that, this is what iPS cells look like. It takes 28 days to turn them into myoblasts, which is kind of this pretty stable, intermediate myogenic progenitor. And then we push them through what we call secondary myogenesis to push them from myoblasts into these really nice myotubes. So for the purpose of today's talk, just appreciate that we can make really good muscle in the dish, okay? So then how do we turn those muscle myofibers into tissues? So we seed them in a trough that has 2 posts. One of the posts is rigid, the other post is flexible. And after we push them from myoblasts into myofibers and pull them out of this casting tray, we then have a skeletal muscle tissue between these posts, and as it contracts, we can measure the force. So what you're seeing in the movie on the bottom is a 1 Hertz broad field electrical stimulation where the muscle contracts synchronously, and then we can measure the force output, okay? I don't need to go over the introduction for Nemaline for this audience, so I think for time, I'm just gonna skip it. All right, so the crux of what I want to present to you is a really productive and enjoyable collaboration with Nigel Lang's team at University of Western Australia, Perth. So Nigel, Rhonda, Gina, and Josh. What their team has done is generate 2 isogenic induced iPS lines from a 4-month-old severe NM patient with a heterozygous dominant mutation of R183S, okay? They did a lot of work to show that they could turn these stem cells into muscle, similar to the way I just showed you, and did some really interesting characterization of the muscle disease phenotype in 2-dimensional culture. So the cells are just grown on flat plastic. And then we teamed up because we were convinced that these 3D engineered muscle tissues would be a better way to measure function, because after treatment, that's what matters to the patients. I mean, you can correct something, engage a target at the molecular level, but if it doesn't lead to improved function and makes the muscle stronger, then that's less relevant. It's less important to the patients that will be treated this way. So we've now teamed up and have done some studies to ask whether their approach of deleting the mutant ACTA1 allele specifically in these iPS-derived skeletal muscle cells actually improves function, okay? So I know this is a very complex slide. What this shows is their CRISPR editing strategy to selectively delete just the mutant allele. So what's shown in the upper left is the R1E3S mutation. The scissors show where this CRISPR enzyme targets this piece of DNA, chops that out, and only the normal allele is left. So, and this is just a lot of molecular validation to prove that it actually worked. But what's directly related to the data I'll show you is, after a lot of cyclic, iterative editing, screening, single cell cloning, what Josh was able to do is to progressively generate cell lines that have more and more of the mutant allele deleted. So the pathological allele is in black, the wild type allele is in gray, and you can see over these selective processes, he was able to generate a line that had 100% of the mutant allele deleted and only the wild type allele left. So that's a really neat comparison. We have mutant or diseased muscle, and then something we think might be phenotypically, functionally rescued. And that's what we put into our engineered muscle tissues. So what's shown here, as I showed you in that movie before, as the muscle contracts, if you give it just a quick electrical pulse, it twitches. So that's one quick instantaneous contraction. So what the folks in my lab, Chang-Ho Chun and Divya Avnur, did was to make these muscle tissues side by side with an N of about 30 per genotype. They put them into this manta ray platform, this two-post contractile platform, and measured them over a month. Every other day, we measured force output every other day for a month. So you can see that as the muscle develops, they really start to stratify. So the mutant, the one containing the mutant allele, is in black, and the one that has been CRISPR edited is in red. And you can see the functional recovery of those muscles when we just give them one electrical stimulation. We also do what's called tetanic contraction. So we give them a longer pulse frequency train. So the muscle contracts and stays contracted for as long as the pulse is present. So again, over this 30-day time course, you can see the muscle, the muscle containing the mutant allele is about 1/10 as strong as the one that has the mutant allele deleted. So this is just selected time points, and I thought it was important to kind of show you the shape of the curve. There's a lot more data that we can extract from the shapes of these curves, what the shape looks like at contraction and at relaxation. I don't have time to talk about that today, but I'd be glad to if you guys have questions. The time course, so we did this 3 different times, 3 replicates, independent experiments, independent differentiations, platings into manta ray, and force frequency curves. And you can see the stratification of the one containing the mutant allele and the one where the mutant allele has been deleted. And again, this force frequency curve shows how muscle functions over the wide range of activities of daily living, and you can see a nice stratification. After we've normalized for the amount of muscle in each one of these tissues, we still see a 2-fold difference after deletion of the mutant allele. Okay? A question we always get is, do you see Nemaline rods in these muscles? And the answer is maybe, okay? Take this with a grain of salt. This is still really preliminary, but let's start with the deleted EMTs on the right. So again, these were all engineered muscle tissues and then sectioned to look for some, with a lot of statistical robustness, we looked at a lot of muscle fibers. So in the instance where the ACTA1 mutant allele has been deleted, we don't see any presence of these inclusions. But in the mutant, with the muscle containing the mutant allele, we do see presence of these kind of punctate staining of titin and actinin. Those are 2 proteins that we think are present in the aggregates. So stay tuned. We think we might have recapitulated an important part of the disease, but it's a little early to say for sure. So the last thing I want to do is talk about the grant that we just got funded through AFBS. Thanks so much for the continued support. We decided to expand what we're doing with multiple different groups. And I think that's one of the real strengths of the foundation is the ability to play kind of orchestra director, is teaming up individuals that have complementary expertise and technology. So we feel very fortunate to be part of that. So what we're going to do, we're going to build on what we've already done, but we're going to try to be a little more clinically focused. For example, in AIM-1, we're going to try to identify the minimum level of pathogenic ACTA1 allele deletion that restores clinically meaningful force. So think about this as a potency assay. Anytime you move toward clinical trial, you need to know how much of your drug or of your treatment leads to how much functional recovery. So we're going to try to establish that deletion function relationship, okay? In the second aim, we're going to identify effective molecular or repurposed drugs that we think could restore function in this platform, but we're going to test them side by side, head to head. So we have a hierarchical ranking by the end of this to be able to see which ones are most promising. And the different categories that we're going to test, we do some molecular modeling in the lab, so we can use these molecular dynamic algorithms to look for chaperones or proteins that help other proteins fold. So if ACTA1 is misfolded, we can add one of these chaperones in the hopes that it helps it fold back to its normal structure. We're going to test that. We're going to test some tRNA therapies that promote read-through to overcome the stop codon to restore normal protein levels. We're going to try to, this is something Nigel's lab has done for many years, but it hasn't been tested in a human functional system. We're going to try to ask again whether upregulation of cardiac actin could be a compensatory mechanism to restore skeletal muscle function. We're going to test Afrooz's CRISPR-H40Y mutation correction that we're going to hear about next from her. I'm excited to hear about progress that she's made. We're excited that that's going to be part of one of the things we're going to test side by side. And with Pete Howling at Melbourne, he has a way to screen small molecules and drugs that we could repurpose for Nemaline. So he has a screening method that he will put these small molecules through using the same platform. And then we're going to work together to make sure that those small molecules work in both of our hands, that we can do dose response curves and things like that. So we're excited about that new collaboration. And we're really thrilled to finally not only focus on ACTA1 and try to establish this same skeletal muscle platform for Nebulin and cofilin mutants. So we're going to work with Julian Ocala's lab. We're going to work with Mary Bailey's lab to get both IPS, so CRISPR engineered mutant lines that we can then turn into muscle. We're going to get Neb mutant patient myoblasts, and we're going to get cofilin mutant cells from Mary to make EMTs. All right, I think that's good enough. Let me thank the folks who actually do the work, and I am glad to take questions.
Thank you so much, David. This was really great. Again, you covered so much, and we do have some questions already. And I think you answered one of them. Will this research be applicable to other genes as well, such as Neb gene mutation?
Yeah, that's the next grant. We have to make the lines first. So we're going to CRISPR engineer them. That takes a few months. And then we'll, whenever you create a new mutant line, there's a lot of work to just characterize that line. Does it go to muscle? What's the disease phenotype look like? What are their molecular perturbations downstream of a Nebulin mutation? So it's probably going to take us a year to just do that characterization. And then we'll put that into our drug validation pipeline.
Great, thank you. And question from Joe Lipsick. Have you tried using CRISPR to edit the R183S mutant allele back to wild type? Some deletions might themselves create dominant inhibitors, as recently shown from some NEB mutant patients.
Yeah, we haven't tried that. We've talked about it. We're focused on the deletion of the mutant allele right now. But yeah, it's on our mind, but I don't have any more to say about it at this point. Yeah.
Okay. The mutant allele appears to grow in strength significantly by day 34. Is that a nod to exercise alone?
So we haven't explored that. Think about, so the way these experiments are done is the muscle will spontaneously contract a little bit in the dish when just left in the incubator. But the more mature the muscle becomes, the less that spontaneous contraction occurs. So when we collect data, it's literally for less than 5 minutes every day or every other day. So basically the muscle is static for most of the time. So one of the things we want to do is put these muscle tissues through an exercise regimen to see if it makes a difference in whether we can see a hypertrophic response and whether it exacerbates the phenotypic defect, because many of these muscle diseases, Duchenne, for example, undergoes contraction-induced injury. So anytime there's a defect somewhere in the muscle fiber, it's quite possible that will we see a hypertrophic response in both tissues, but will the exercise, as we alter the frequency, the duration, the strength of the electrical stimulation, and how much rest time we give them between these bouts of exercise, will that help improve the stratification? We don't know yet, but that's one of the things that's in a different grant that we're pursuing.
Okay, great. I'm going to read a comment or question from Hank Granzier, which I'm so happy to see that we have a comment.
Good to see you, Hank. Thanks for coming.
Yeah, we have families, affected families, and researchers on the call today. So great to have you, Hank. Hank says, great talk, David. How close is specific force of CRISPR-corrected EMTs to that of WT EMTs?
Yeah, so that's asking how close. So we have diseased rescued, but we don't have the comparison of wild type because these are patient-derived cells. We don't have the wild type comparator with the exact same genetic background. So Hank, that's what we're doing. We created the H40Y mutation by CRISPR. So we have the normal CRISPR mutant H40Y, and then we're gonna do the H40Y correction, and then test the various therapeutic interventions as well. So we'll have that comparison, but we can't really do that with the patient cells.
Okay. Another question from Howard. Have you used CRISPR successes from other diseases, including dystrophies, to help shorten your research pathway?
Yeah, I don't know if you're aware, but most CRISPR strategies work well in isolated cells or tissues in the dish, but at the clinical level, it's still an enormous delivery problem. I mean, muscle makes up 40% of your body. So getting the CRISPR editing machinery into all the muscle cells is still the major challenge. We are working on several other muscle diseases that we work closely with Jeff Chamberlain's lab and Nick Bengston, who's a CRISPR base editor guru. So we're doing this a lot. How well it works in our engineered muscle tissues is something we're just starting to do. And whether or not there's so many different CRISPR editing strategies out there right now, whether or not they all work equally well in the human tissues, as has been shown in animal models, and whether one is particularly better or worse in human cells, we don't know that yet, but we're working on that.
Okay, and then one more question. I think that, so the question from Jamie, was it tried with TNNT1? I believe maybe the
No.
3.
No, not yet, but I want to give a plug to one of the things I really admire about AFBS is as soon as you've made let me do it this way. If you want to study a new disease or a new mutation, you can't get grant funding from the NIH until you've made the line, characterized it, shown that there's a disease phenotype, and that there's something there worth studying. So somebody has to have the vision to take the chance to say that we're going to give you some money to just do it from scratch. The reason I think that's important is because what we do can be driven a lot by what we get funded to do. So for us, making an additional mutation from which we can make muscle tissues, it's $10,000 to $15,000 to make a line, and about 4 to 5 months. So in other words, it's not a heavy lift to include other mutations, but the characterization of each individual new line is a full-time job for one trainee. So this is not something that can be an add-on, kind of this additional project that somebody takes on. This is a lot of work. So if we work together as a community to decide what other mutations should be studied, then we just do it, and we have the support of a trainee to make this their life's work, and we just do it.
Thanks, David. I appreciate you sharing that and explaining the complex process that we're facing, and the progress we're making towards, achieving these goals.
Yeah,
Challenging, but we're making great progress, and it's very exciting to see what you're doing. A pleasure to have you here today. Thank you for your time.
And I'm looking forward to hearing from Afrooz.
I am as well. So we will pass it right over to Dr. Afrooz Rashnonejad from Nationwide Children's Hospital. Welcome.
Hello, everyone, and thank you for the invitation. I'm so happy to be among you today and for all the supports for our grants and work. And as like David said, it's impossible to do this works without you. Okay, so can you see my slides?
Yes.
Perfect. So today I will talk about my project updates on gene therapy development for ACTA1 patients. And I will pass this slides. I know you are familiar with this disorder, but very quickly want to mention that this is one of the subtypes of Nemaline myopathy that caused by many mutation. Over 100 different mutation has been detected till now in ACTA1 gene, that is alpha skeletal muscle gene, actin-1 gene. And majority of these mutations are inherited in autosomal dominant, like 90% of them. And so there are lots of de novo mutation that maybe parents, they don't carry these mutations, but the kid will have that. So it's a mix of inherited plus de novo autosomal dominant mutation, and accumulation of this mutant activin proteins in like the muscle cells can cause the phenotype and symptoms of muscle atrophy and muscle weakness, and finally lead to the death of the patient or wheelchair dependent. So we are using the AAV-based system to cure or treat this genetic disorder. And we are using adeno-associated viral vectors. I always show this slide when I'm talking about the adeno-associated viral vectors. These are very tiny. If you can see them at the bottom, like corner, the left corner of the figure, that they are very small viruses, only carry 2 genes that they are non-pathogenic. So we technically take out those 2 genes and replace them with a therapeutic gene, and then make them in billion particles, concentrate them, and then treat or patients with them. And so as you see, they are smallest viruses, like compared to coronavirus, for example, 50 times smaller than coronavirus, or about 20, 30 times smaller than bacteriophages or other viruses, and compared to other cells in our body, like lymphocytes or red blood cells. So you can think the AAV vectors as a Trojan horse. So how this gift brought those soldiers to the city and released them to fight for them. So exactly, the AAV vector does the same thing. This capsid proteins that you see in green and reds, they have receptors for the cells that they're targeting to enter into the cells, to the target cells. And they carry a therapeutic protein. So if you remember, we said that none of the AAV vector genes remain in this vectors that we make. They are 100% replaced with the therapeutic gene, right? So we make lots of them and then use them for gene therapy. Today, there are about 7 AAV-based gene therapies approved by FDA, and they are available for treating different disorders, including the first one that has been approved in 2017 called 𝗥𝗲𝗀𝗃 for the inherited blindness, and then 𝗔𝗙𝗕𝗏 for spinal muscular atrophy, or Roctavian for like factor VIII deficiency, and other one, and 𝗨𝗟𝗡 that like 2 years ago has been FDA approved for DMD disorder, and that's the AAV-RH74. So the 𝗔𝗙𝗕𝗏 and 𝗨𝗟𝗡, like 2 AAV-based gene therapy out of this 7 has been approved, and like this 2 has been developed at the Center for Gene Therapy at Nationwide Children's Hospital, where my lab is located. And you can look at the cost. So they are pretty expensive costs. But the good point with this AAV-based gene therapy is that they are one-time treatment. So when you're treating the patients with this gene therapies, you expecting over 15 years. And technically it says for long term, like lifelong expression of the transgene. So you don't need to get this gene therapies again and again. And the strategy that we are using for activin-related Nemaline myopathy, or NEM3, it's I can make it a little simple to understand. So think that you got a recipe from a internet, from ChatGPT, for making a cake, and you bake that cake, you try it, and oh, it's salty. So instead of having enough sugar in that recipe, it had more salt or something. The recipe wasn't looking good, so there was something wrong with that. So you like toss that, you trash that recipe, and you get a wrong one from a trusted person, from your mom, from your friend, and bake the correct one. So correct amount of the sugar to have this delicious cake. So our strategy is exactly the same thing. Our bodies received wrong genes, or has for any reason a defect gene for activin and has that mutation, and they are making a bad protein that it's not functional, and actually it's interfere with even right proteins functions. So our strategy is to use a small RNA called microRNA to clean this bad gene or messenger RNA from our body. You see, let me just turn my laser pointer here. Oops, wait. Okay, so you see the microRNAs in red. They are very short RNA that we design them specifically to target the active one. And then they go and they find the defect active one and they digest them. So you see these pieces in blue. So they are going in the trash in our cells. And then at the same time in that AAV vector, we have a healthy copy of active one gene that can produce a healthy active one protein that is actually functional and can do its work in our muscle cells. So we designed several microRNA. I showed the structure of those microRNAs here. We made several of them, 12 of them, and we tested them in vitro in some cells that had overexpression of the healthy wild type and 2 mutant activin, H42 and D288G variants of activin. And we found that 3 of them were very successful to reducing the expression of bad activin in our body. And then when we made another copy of healthy activin that was resistant to be knocked down and silenced by this microRNA. So you see here, this is the normal wild type or mutant activin. And then we changed some of this like nucleotides in a way that you still coding the same amino acids. So see the PSTM, the reading of the amino acid at top, for example, strand is similar to the lower one. So you have the same amino acids, but it's resistant to the microRNA activity because the microRNA cannot 100% like align to that one because of this mismatches. So this would be the healthy messenger RNA for active one that we put into AAV alongside with the microRNA. So what we did, after making this 2, we made an AAV vector that was carrying the microRNA first, and we tested that in vivo in our co-injected mice tissue that they received a wild type or the H42 activin fused to the GFP or D288G, a variant of activin fused to the GFP. So this GFP acts as a like reporter for you. So this is fluorescent protein, and if you inject the muscle, you get lots of that like mutant protein that it's fluorescent in green. And when you're treating them with those 3 microRNAs that we have here, like either of those microRNA that pack into AAV vectors, you see how successfully we clean those variants from the muscles. And when you look at closer to the sections of those treated muscles, you see that they are completely gone. So those bad active ones are gone. So then we packed our AAV vectors with both microRNA that we selected, the lead microRNA, and also the lead construct for making healthy activin protein. And we tested that in vivo and in vitro, and we found that we can reduce the expression of the endogenous activin, but the resistant, like this is the resistant activin that can be expressed without reduction by any microRNA. After that, our goal was to testing this gene therapy in animal model that we have. And the best animal model currently available is the age42 animal model that like has showing muscle atrophy, aggregates of mutant ACTO1 in the muscles. Also in like the graph A, you see the reduced body weight by time, and affected that they are in red compared to the healthy litter mates that they are in blue. And then the functional assay that it's grip strength, so that's the assay we put mouse on a mesh that it's attached to the machine that measure the muscle force, and we drag the tail of the mouse so they will drag the mesh toward them, and we measure how strong their muscles are. So this measurement showed that almost all the untreated affected ACTO1 animals, they have significantly lower grip strength or muscle force compared to the healthy wild type litter mates. And then the survival was much more affected compared to the healthy ones. So our goal was to treat mice. Pre-symptomatic means immediately after birth, and then during the like adult phase, it's about 4 to 8 weeks of age in mice, and see how our gene therapy works. So just wanted to quickly show the construct of the AAV vector that we have. As you see here, we have a muscle-specific promoter. So it will, the expression of our ACTO1, because it's very important to have expression in the muscles, it's limited to only muscle. So not other cell types or tissues can express that one. And it's tagged with the very tiny protein called FLAG that helps us to differentiate the transgene that we replace compared to endogenous, like bad ACTO1. So we can measure a ratio of the how much endogenous bad mutant ACTO1 we have in the cells and how much of this good healthy ACTO1 we are replacing using this FLAG tag that differentiate them from each other. And then we have another construct that has its own promoter and expressing the lead microRNA to knock down the endogenous ACTO1, and we pack them into AAV vectors. And as I mentioned, the goal is to treat pre-symptomatic and post-symptomatic mice. So in our first trial, preclinical assays that we did, and this is very low numbers of the mice, so we need to increase these numbers to get very meaningful statistical values between treated and untreated, and do lots of assays. This is very recent data that we got. So we look at the body weight of the healthy animals, either treated or not treated with the AAV vector gene therapy that we have. And then we look at the H40Y animals that they are affected, but they didn't receive the gene therapy. And then those that they received the gene therapy. So the body weight didn't change that much when we look at that at 6 weeks. But we need to continue that to them because 6 weeks is really early, and this mice showing most of the phenotypes after 13 weeks of age. So we need to keep them going and do the analysis on like longer age, longer time points. And then we look at the grip strength. The grip strength or muscle force was very obvious outcome for these animals that has been published many times. So we believe that we should trust the grip strength as an outcome measure if we want to look at gene therapy. And as you see, the treated group at 6 weeks of age demonstrated significantly increase in muscle force compared to untreated animals, but still it's lower than like healthy, unaffected animals. But as I mentioned, this is very first experiments that we did using our gene therapy, and we need to optimize that, the dose, the route of delivery. This is one dose we tested, almost like very medium dose that we wanted to see how much recovery we can get using that medium dose. And then we continue this study without sacrificing mice. As I mentioned, when these mice are aged, the phenotype are more severe, and you can see more phenotype in these mice. So we have different groups injected in different, like these are from a batch of litters born from like one single parents, you know, and they're born in a different time points. For 6 weeks, we had all of them done already, but for 16 weeks we only did that for a couple of the like litters, and we need to keep that for rest of them to have the same numbers that you saw in a previous slide. So for now, this is what we have for 16 weeks, and we saw more improvements in treated animals closer to wild types, and more reduction in grip strength and H40Y. As you see, this is the first batch, so we need to continue these measurements for the rest of the injected animals and add them to this group when they reach out to 16 weeks of age. So this study is ongoing, and these results are very fresh. We just presented those results last week to Alan Beck's group that we had the joint lab meeting, and this study is still ongoing. And the next phase from like those preclinical studies has been funded recently by AFBS. So we hope that inject much more mice pre-symptomatically and post-symptomatically, and look for histological outcomes, and also behavioral outcomes, grip strength, and survival, and other measurements to show the efficacy of our gene therapy and the safety at the same time. And we also, in another aim of that grant, the very recent grant, we will generate a like cardiomyocytes or cardiomyocytes lines from the patient fibroblasts that we converted them first to the iPSCs. So in the left, you see patient cells, the fibroblasts that we convert to iPSCs and perform the lots of assays like the embryonic proteins or pluripotency markers like OCT4, SOX2, to make sure that they are really iPSCs. And then we differentiate them into the muscle cells and perform the muscle like measurements to see how the force of this muscle is, because a portion of active and patient, they show the cardiac issues, either the hypertrophy or dilated cardiomyopathy. So in collaboration with a cardiologist in our research institute, we are making the cells, and we will look for disease mechanism, because we know that activin is very important protein in skeletal muscles for our movement. And but why sometimes heart is affected in this patient? Because our gene therapy is a universal gene therapy, and we believe we can treat all the patients with any mutations in activin, so either with cardiac phenotype or without cardiac phenotype. So we want to see what is the mechanism in those patients, and if our gene therapy can treat them as well. So with that, I would like to thank all people in my lab at Nationwide Children's Hospital, and also our collaborator, David Mack, Kevin, Alan Beggs, and others who help us to do all these assays. Unfortunately, I wasn't able to show the CRISPR results, but here I just focused on AAV-based gene therapy because of the time. So thank you so much. I will stop sharing my slide and would be happy to answer any questions you have.
Thank you so much, Afrooz. Great information, incredible work you're doing. First, we have a comment from David Mack. He loves your AAV slide comparison, so if you can share that with him, that would be great.
Of course.
It is a great slide.
Of course.
And let's see, from Carolyn, how long do you expect it will be until human trials?
So we hope soon, but we need to do lots of experiments and show our gene therapy is really safe at the same time that it's efficient for patient, but it's safe. So safety is always the biggest concern with the gene therapies. We don't want to but adding something like bad additional to our patients. So we're caring about that part a lot. But generally with this all advancement in the field, it's much more faster. So if in the past the timeline was like 10 to 15 years for a treatment to go to the clinic, now it's much more faster. But as David mentioned, really the matter is funding. So you see today how unfortunately like some of this cuts happen in NIH funds, and then like it affects the biomedical research and also treatment development for patients. So if there are not enough money to showing the efficacy, if there is not enough money to show the tox in large animals like in pigs, in non-human primates, so it will delay our progress to the clinic, you know. And I hope things get better. But again, it all depends on how much like support we can receive. And I want to mention we are very thankful for our patient community to support this research. Without the research, it was impossible to make this work happen. And again, as David mentioned, I'm just emphasizing what he said. So it's impossible to get NIH grant with no data. So you need to first get some funding for your preliminary work through the foundations, through the monies that are donated to the foundations, and then make some data and like show your reviewers that you are capable of doing this research. And then you can get like $1 million from NIH to accelerate your research.
Thank you, Afrooz. We're grateful that we're able to support this work. And there's another question from Joe who says, very nice work. What do you think of the problem with Sarepta AAV therapies for Duchenne MD mean for the field? So I cannot talk for the Sarepta because I was not involved in that research. Although it has been the research has been done at the Nationwide Children's Hospital, but it has been done by a separate group, so I cannot talk for them. But the biggest thing is the microdystrophin itself may not be that efficient protein compared to the full-length dystrophin, and they need to and the vector that they are using is AAV-RH74, so they needed to use very high dose of that one systemically. But today, with the advance in the field, having muscle specific AAVs like MyoAAV that transduced muscle cells very efficiently with very low, like lower doses, like 10 times lower doses compared to other AAV vectors. You can reduce that dose and you can reduce the side effects. Like if, for example,
there was liver tox with other AAV vectors, AAV9 or AAV-RH10. Now, by reducing the AAV dose using myo-AAVs, you can prevent that liver tox. So that could be preventing. And then again, like Jeff Chamberlain group is doing an excellent work to making a full length dystrophin and then using a muscle tropic AAV to doing two things at the same time, transferring a full length protein that is more functional and then having a muscle specific AAV so they can use a lower dose. And by doing this too, they can prevent those issues that we may saw for microdystrophin.
Thank you. Arash has a question about any upcoming trials for AAV that they can join for late onset NM.
Well,
Not
is that
yet.
specific? Yeah. If that's specific to ACT1, I can answer that there is nothing yet. But for other like NEM disorders, I'm not aware, but I don't think so, right, Dave? Yeah.
Yeah, not that I know of.
Yeah.
Yeah.
Let's see. Okay, we have, I'm curious to know if you have strategies. Are you exploring to enhance transduction efficiency while minimizing immune responses in AAV-based therapies?
Yeah.
Yes, this would be a very latest step of our gene therapy development. So we are now in very early step of showing that the principle is correct and we can do the knockdown replace and we can reverse the disease phenotype. And after that, we can look for immune responses and things against the AAV serotypes, but this would be very later stage, like in future. Yeah, not in this current study.
Well, Afrooz, you already did this, but I wish I had a better estimate of the amount of money that has been put into developing better AAV muscle targeting vectors, these so-called myogenic AAVs. So 𝗕𝗏𝗏𝗏, the Myo AAV series, Dirk Grimm, ironically, the AAV Myo series. They're much better. It's a little bit in dispute how much better. Is it twofold, fivefold, tenfold? But many companies are putting huge amounts of money into this. So they have freedom to operate on a unique vector that they can then control. So stay tuned. It's going to change the field. These new vectors are going to change gene therapy.
Great. We're going to take one more question and then our time is going to be wrapping up here. We have a question from Stacy. If human trials are successful, is this treatment meant to be used if detected in utero, or is there expectation for affected individuals to improve? Are there any reasons why someone would not be a candidate for treatment? It looks like, did we lose Afrooz? I don't see her on here.
Oh, wow.
Yeah, she's gone.
Oh, shoot. Well, Gus, any thoughts or David?
David? I can, of course, I can give my
Yeah,
opinion
please.
is,
We
yeah, I mean,
heard. Yeah, please.
yeah, this research is for doing these studies not in utero, in living individuals. So, and the range of ages is something that we don't know yet for ACTA1, or for Nemaline myopathies in general. There is this principle that the sooner the better, and this works for the NEM also. But the field thinks that even in adults receiving this type of therapies, at least they can stop the progression of the disease and improve. And of course, as older as you are heavier and you need more of the virus vector, which is potentially more toxic. So there are many things to tune up, but no, the idea is to treat patients in all age ranges.
Yeah, so I'll just add, that was great, Gus. I'll just add, in almost every meeting we go to now, whether that's MDA or ASGCT, the American Society of Gene and Cell Therapy, in utero delivery kind of gets at least one session, so people are really starting to look at it. And this is obvious, but it's worth mentioning. Babies don't have an immune system yet. So whatever immune response that may tamp down the ability of the vector to stay around longer and to deliver the cargo could greatly enhance transduction. And Gus already hit upon the fact that, the babies would require a much lower dose, and that helps with manufacturing, and it would be highly desirable if we can work out whether or not the dose that the mother gets might be deleterious.
Hello again. So sorry to be disconnected. My internet has problems. I tried to just somehow solve that issue and got reconnected. Sorry, Dave, I didn't hear your question, but I can imagine that it was related to the Mayo AAVs and maybe.
Yeah, I tried to cover that. You're more of an expert than I am, but I did my best.
No, thank you. Thank you for covering that.
Well, do you have any comments on in utero delivery?
In utero? Well, my lab is working on in utero gene therapy.
I thought you were, yeah.
Yeah, we published a paper for SMA in utero gene therapy in 2019, and now we are trying to optimize the protocol for many other neuromuscular and neurological disorders that they are starting in utero, so you can prevent those phenotype much earlier before birth. And as you mentioned, for the babies and in utero, like embryos, you will need only a portion of that AAV that you need for treating an adult patient. So technically, the cost is much more lower, and then because the dose is lower as well, and the baby doesn't have that immune responses, so you can treat them with better results, and lower cost, better results, less immune response, and sometimes immune tolerance to that transgene, for example, like dystrophin, that you transfer before birth. And yeah, we are trying to optimize that now in the lab for very severe early onset neurological and neuromuscular disorders.
Nice.
Well, we have hit our time, and so I want to just I know we could go on for probably hours more with questions and conversation about this. There's so much to talk about and so much still to understand and discover, but we're really, really grateful for you, David, and Afrooz and Tina, to be here today and to share the work that you're doing. It's so critical to help us get closer to treatments for NM. So just from the bottom of our hearts, thank you for all of your hard work. We're honored to be able to support you from AFBS, and just keep up the great work. One thing to Afrooz's note about, and to David's note about the importance of funding, AFBS is funded by donors, individual donors. And so we do have a special announcement. You'll hear it here first before we announce it on Monday, but we have a group of donors that will be matching up to $100,000 this year. In the past, it's been 50. Now we have $100,000 to match. So up and through the end of this year. So I just highly encourage everybody to donate if you can, or spread the word to get donations or fundraise with your network, because because none of this research would be possible without your donations. So I think it's just important, essential to say that because we need to have money in order to fund these important projects and get us closer to finding a treatment. So we're really grateful to have you guys. We would love to have more sessions like this in the future. So we'll plan to feature even more of our researchers that we're funding. In 2026, we'll plan to have another one of these. I want to thank everybody who has attended today and taken the time to listen and learn. Please reach out if you have any questions, which I'm sure you will. There are so many questions and so much still to learn, but AFBS is here to support you in any way we can. So thank you so much, and I hope you have a great rest of your day.
Thank
I
you
want
so
to say
much. It was
thank you
awesome.
to everybody. And if you have any question, please send an email to us. So we will be happy to answer your questions.